The second presentation will be given by Jordi Petriz from Barcelona, and he is a very big expert in cancer stem cells and in cell populations. And you will see in a moment what he will tell us. Jordi, yours. You want to have this one without the wire?
Okay, and thank you very much for this nice introduction, Andrea, and thank you very much for inviting me for this very exciting meeting. I would like to say that I'm overwhelmed by the quality of the speakers, and then I would also like to thank Thermo Fisher, and especially Jolene Bradford, Mike Ward, and Paola Paya for their continuous support, which is fundamental for the work I am doing at the Josep Carreras Leukemia Research Institute. Josep Carreras, as you may know, is a tenor who suffered leukemia and was cured, and he decided to start research in the specific field of leukemia. And my institute is mainly organized by 3 different campuses in Barcelona, and these 3 different campuses are mainly composed by the 3 main hospitals in my city. Oops. Okay, my first steps in the field of flow cytometry were related to the cell cycle and ploidy analysis, and also I was very interested in the study of CD34 positive cells using flow cytometry. And after these first steps, I was very interested in the expression of multi-drug transporters, which are expressed in stem cells, but are also expressed in cancer and leukemia. And my PhD was in the field of the functional study of the activity of the P-glycoprotein, which is not only expressed in CD34 positive cells, but also in cancer and leukemic stem cells. Using this expertise, I was able to learn more about the stem cell compartment, and then we started some experiments to learn more about the stem cells, especially about the CD34 negative compartment. And this is the summary, the field I'm studying, and by combining these 2 last steps, the stem cells and CD34 negative stem cells, we study the activity of the ABCG2 transporter expressed on tight population cells. And finally, we are very interested in a very exciting field, which is commonly termed hypoxia, but I would like to say that I prefer the term physioxia instead of hypoxia, because hypoxia is the normal concentration in cell tissues. This is a classical distribution, or the classical scheme of hematopoiesis. Oops. Sorry, because I'm missing the button. Oh, okay. This is the classical scheme for the hematopoietic compartment and cell differentiation. And this is the blood stem cells, which is able to self-renew and to differentiate into different progenitors and to different cell lineages, such as red blood cells, platelets, and white blood cells. And we are looking at very rare cells. Maybe the hematopoietic stem cell is the better studied model, and we know that they are able to express multiple markers, and these markers are useful to study the normal stem cell compartment. And some other markers has been developed to study different leukemic or cancer stem cells. So there are multiple publications in the literature, talking about the expression of CD44 and other markers, and we are very interested in the combination of phenotype and function to learn more about the stem cell compartment. In 2000, a paper published in Cell discussed the possibility that the stem cells can be a function instead of a phenotype, and most most stem cells can, or stem cells from different tissues can express mature and markers from undifferentiated cells. And then we were initially very interested in the CD34 positive marker, and we are now studying different markers I would like to discuss during my talk. We need to understand that if we are dealing with the stem cells, we are looking at rare cells, but there are also some classical stem cell markers that I consider, in my experience, that are not good, and this can be the worst stem cell markers, such as the BRDU, because it's not specific, and in addition can be toxic, and more than the 99% of cells that retain BRDU are not stem cells. OCT4, which is an embryo stem cell marker, is only for the young, and OCT4 levels in adult stem cells, well, are very different when compared with adult stem cells when embryonic stem cells. And CD133 can be apparently expressed in gliomas and astrocytomas, and we were unable to obtain a reproducible staining system when this marker was used in my lab under these two models. And if we are looking to the presence of rare cells, which are less than 10 per microliter, sampling will be very important as sample preparation. Perturbation of the assay is also very important as the counting statistics. Hydrodynamic, the classical approach for flow cytometric studies, and acoustic focusing, which is a new technology, allows for a better rare cell detection in our experience. Data processing, raw data analysis, subset analysis, this is the combination of multiple parameters. Antigenic density, which may vary between the different stem cells from different tissues, or some problems associated with flow cytometry and other techniques such as the carryover, the system noise, or the fluidic stability, which was commented previously by Enrique, is fundamental. And some other techniques, such as the pre-enrichment techniques, are also crucial to study these rare cells. And this is an marrow aspiration, and if we are looking at the presence of then 10 cells per less than 10 cells per milliliter, we need to know that we need a huge amount of cells, because these results can be significant for our studies. If not, we will have a problem with sampling, and in the case of bone marrow and leukapheresis products, which are highly concentrated products, we need to know that a minimum amount of cells that we should test in our laboratory is needed. Well, this slide is dedicated to Jolene Bradford, because I know I love an special animal, but if you are looking to this slide, we need to know that if we are looking to rare cells, we can also detect some artifacts, and maybe someone in the audience will be prone to identify the rare stem cell or the rare event present in this slide? No? Okay. This is a cat, this is not a penguin. Okay. This is a modification I performed in my computer to show that if we are looking at very rare cells, some artifacts can emerge, and this can complicate the issue. And we are also observing rare events, but also in both space and time. And this is unstained HeLa cells undergoing cell division, and this is a case on a rare event, and one cell is dividing into multiple cells, but this is time depending. And if we are looking to this rare event, we need to know that time is also crucial to observe these special cells. Okay? And I think that we need to think in absolute counts. We need to forget a classical parameter used in flow cytometry, which is percentage, and we need to focus in the populations that we are looking for. If we are using most of the studies I'm performing in my lab uses whole blood, as is the studies that Enrique explained previously. And this is a classical pattern of unlicensed whole blood, and leukocytes appear as the rare populations. And main populations are erythrocytes and, secondly, platelets. And then if we are looking to stem cells which are contained in this compartment, we need to work with very stable and high throughput technologies to ensure that we are detecting the good cells and not the cat between the penguins. Okay? And this is to summarize that this, the detection, for the detection in blood and everything counts. And this represents the cell number, and this represents the cell type. And if we are looking for circulating cancer stem cells, these cells can be in a relationship from 1 to 1 liter of blood, for example. And this is very complicated because we cannot sample this. We are unable to sampling this large amount of blood to detect circulating cancer stem cells. For circulating tumor stem cells is different because we can detect 10 from 10 milliliters of blood, but we need to know that numbers are crucial. Not only the technology, but numbers for sampling are crucial. Okay? And then remember that in function, it will depend of the number of the population that we are looking for instance, in blood, the numbers that will represent and the sampling that we will need to perform. Well, this is an old paper that I published in cooperation. These experiments were done at the Cancer Research Institute in Barcelona, and this is a very simple triple staining to identify very primitive progenitors. In this first plot, we stain the cell. We discriminate the necrotic cells using 7AAD, and this is a cluster of CD34 positive cells that were stained with a specific antibody conjugated to phycoerythrin, and these cells were stained with rhodamine 123, and this is time, and this is rhodamine 123 fluorescence. And we register for the first seconds the autofluorescence of the CD34 positive cells. The Boolean gating is represented here. Viable, non-necrotic CD34 positive cells represented here. And then there is a lapse to add rhodamine 1 to 3 to the cells. And then we can monitor the mitochondrial membrane potential. As you will see, most of cells of CD34 positive cells have a higher mitochondrial membrane potential. In addition, there is a few population of CD34 positive cells with an increased fluorescence for rhodamine 1 to 3, but a minor fraction of these cells can also express low levels of not express. You can detect very low levels of rhodamine 1 to 3 because of the low membrane potential, but also because of the expression of P-glycoprotein in CD34 positive cells. Rhodamine 1 to 3 is one specific substrate for P-glycoprotein, a multi-drug transporter, but also for different multi-drug transporters. And this is a simple triple staining assay to distinguish very primitive CD34 positive cells, because based on the membrane potential activity or the mitochondrial membrane potential, these cells are more quiescent when compared with the more brighter cells. And in addition, it was published later in Cell that the most primitive CD34 positive cells also express the highest levels of P-glycoprotein, explaining that this low staining for rhodamine 1 to 3. The classical primitive stem cell compartment has been also described by the combination of two markers, CD34 and CD38, and the most primitive cells are enriched in the CD38 negative compartment. And this is a comparison of different 6 specimens, bone marrow human specimens, showing that this compartment is quite different when you compare different specimens. And we also need to take into account that if we are manipulating mice or if we are looking at the presence of these cells in humans, will vary more in comparison with the mouse model. And in addition, stem cells are classically defined as more quiescent cells compared with the more differentiated compartment. And this is a triple staining experiment. And then we sorted the cells because at this time we were unable to simultaneously stain the DNA in combination with these 3 markers. This was done more than 20 years ago. And if we look at the cell cycle of the CD34 positive, CD38 positive, DR positive cells, we can see that the 83% of cells are in the G0, G1 phase. Near the 15% of cells are in S phase, and near the 2% of cells are in the G2M phase. If we look into a more primitive compartment, because they lack the DR marker, we can see that G0, G1 cells are 96% of cells. Only we can find the 3% of cells in the S phase, and less than 1% of cells are in G2M phase. And going back to a more primitive phenotype, which is CD34 positive, DR negative, and CD38 negative, we can see that most of cells are in the G0, G1 compartment, the 99%, and only the S plus the G2M phase is near the 1% of cells, indicating that very primitive stem cells are quiescent. But using the classical staining using propylium iodide, we are unable to distinguish the G0, G1, the G0 from the G1 phase. And if we work with enriched cells that were mobilized with GCSF and were selected by immunomagnetic techniques, we can distinguish by combining PARNY, which is an stain specific for the RNA, and the H dye, which is specific for the DNA. We can compare the RNA high, which is mainly enriched in G1 cells, and the RNA low compartment is mainly enriched in G0 cells. And then this combination allows to distinguish also a more primitive compartment when we are looking to the stem cells. This was applied to the CD34 positive cells, but it can be applied to different models. And in addition, this is also an old paper, we resorted to use a very nice and elegant technique, which is the flow fish, to study or to detect a rare fraction of human hematopoietic stem cells with large and extremely large telomeres. And we also sorted this different population, showing that cells with large and extremely large telomeres have a special staining for and a specific location in the nuclei of the telomere. And the more the cells with very large telomeres, when we sorted these cells, we saw that we watch a specific pattern, maybe related to heterochromatin distribution, indicating that this can be a deep stem cell model that can exist into the CD34 positive compartment. And this is a very nice approach to study more primitive cells. But the problem is that you need to kill the cells to study the telomere length in these models. In addition, and this Enrique was talking previously about the perturbation of the assay. This is the principle of Heisenberg. And we also avoid sample manipulation. We also resort to use the minimal sample manipulation, because this can also perturbate the assay, and this can provoke that some artifacts can appear. And this is a classical experiment to compare the effect of red cell lysing by combining a stain that can be used to distinguish lymphocytes, monocytes, and neutrophils. This is dicycle violet, which is a permanent and viable stain that can be used for immunophenotyping analysis to avoid lysis. And when we lyse the cells using ammonium chloride, which is the most common solution, we can count for this number of cells. And if we resort to avoid this red cell lysing, we can see that most of cells were depleted when we compare the lysing with the non-lysing method, and we prefer to use this non-lysing system to detect very rare stem cells. Not also the CD34 population, but also the CD34 negative population, but also to study the presence of cancer or circulating stem cells or leukemic stem cells. The exclusion of coincident events is also very important, and because using flow cytometry, we need to distinguish single cells from non-single cells, and we can distinguish. This is an example to compare coincident event and without coincident event. And if we want to look at these rare populations, we need also to distinguish, to prepare this, to design our experiments using this classical coincident event discrimination. There is one additional method that was described in cytometry a long time ago, but not using the violet lasers. If not, maybe Paul will remember this article. I think that these experiments were done using red laser excitation, but this is a classical experiment that we perform in our laboratory using the blue side scatter and the violet laser side scatter to distinguish platelets, leukocytes, and erythrocytes. And this is a different system to distinguish these populations in analyzed whole blood, and we also perform the CD34 counting using this nice discrimination. And the only that we need is add a threshold in the fluorescence for the CD45 antibody, which is conjugated to fluorescein. This is a leukocytary marker, and as you can see here, this is the analyzed pattern of the leukocytes and monocytes and neutrophils. This is the forward and side scatter distribution of analyzed cells, and we can monitor here the presence of CD34 positive cells, as you can see here and as you can see here, but avoiding the classical red cell lysing approach. In addition, you can add your dicycle violet or any other nucleic acid stain to distinguish the red cells, analyzed red cells from leukocytes. And as you can see here, we can only by simply adding the dicycle violet distinguish lymphocytes, monocytes, and neutrophils. This was done to distinguish, to gate the singlet population, and this is a classical distribution of lymphocytes, monocytes, and neutrophils. And here, this is very tiny, there's not much brag, but you can see here the CD34 positive compartment. In addition, if you have multiple lasers in your lab, you can enhance the resolution of the populations that you are looking for. This is a case that we compared the blue excitation of the CD34 marker and the CD45 marker in a leukemic specimen. In our hematology department, they are using this cytometer, and this is a classical panel combination using these markers PC7 and ECD for the identification of these two populations. But using the classical staining with 5-correctorin conjugated CD34 antibody and FITC CD45 conjugated antibody, we can distinguish, we can compare the distribution of these cells, but using the blue and yellow excitation. We don't need color compensation, and in addition, we can distinguish this lineage, maybe these stem cells, leukemic stem cells, or this blastic leukemic population was originated in the CD30 negative and CD45 negative compartment, and we are unable to compare this nice distribution with a classical staining using the blue excitation. In addition, there is a problem when we are looking at the stem cells, because there is a subjectivity in flow cytometric measurements, and we performed a long time ago 3 different trials in Spain and Portugal to compare how the different centers that are involved in the clinical measurement of CD34 positive events are measuring this population. And the third trial consists in send to the different laboratories FCS files, and then only using their own software to count the CD34 positive cells, showing that there is a huge variation in the number of cells. This is a coefficient of variation for the different laboratories, showing that there is a huge subjectivity in the CD34 positive measurements that will affect also the rare cell measurements if you are looking to cancer or to leukemic stem cells. Okay, and then we need to know that if we are basing our experiments in functional flow cytometric measurements, and we are looking through the 2 main transporters that are involved in stem cells, the P glycoprotein or MDR1, which is termed ABCB1, is mainly expressed in CD34 positive cells, and the ABCG2 transporter, which is responsible to the resistance of mitochondrion and was described in highly resistant breast cancer cells, are mainly expressed in the site population, which are CD34 negative cells. These multi-drug transporters have some physiological roles, and we believe that, I will explain this later, that they act as firewalls for transaction signaling, and I will explain the main reason to our hypothesis. And these are very complex molecules, which some domains for the binding of ATP, and by ATP consumption they are able to efflux some drugs and components from the inside to outside of the cell. And ABCG2, the molecule that is expressed in site population cells, seems to be an homodimer and needs another homodimer to be functional. Can you turn down the lights? Because this is to show you how we engineered a molecule in our laboratory by combining the green fluorescent protein and the pink glycoprotein. Oops. No. Well, but these cells. This is a pool of highly resistant cells because they express a highly amount of. No worries. I think that I can explain that with this powerful light. This is the protein, the chimeric protein that we designed and we overexpressed in our highly drug-resistant model. How to show how the staining is mainly located in the cell membrane, but also we can see how the cytoplasmic levels can also be detected because they are producing the active pump conjugated to the enhanced green fluorescent protein. And these cells were co-incubated in the presence of a chemotherapeutic agent, which is also fluorescent, which is doxorubicin. Doxorubicin makes ternary complexes with the DNA topoisomerases and the proper and the molecule. And as you can see here, the highly resistant cells are not accumulating doxorubicin in the nuclei because they are able to efflux actively this molecule, this drug, outside the cells. And the cells with a lower transport expression have an increased staining of the nuclei, and then we are producing damage into the cells. And as you can see here, most of cells lack the P-glycoprotein, and they accumulate the doxorubicin, and this will provoke the damage, and this will kill the cell. This will kill the cells. However, in many occasions, chemotherapy is killing the vast majority of cells, but acts by selecting this population. And this is very common when we look at or when we resort to use some taxanes or some other molecules that are blocking cell division, because these molecules also have the capacity to transactivate the mutant p53 protein, and mutant p53 protein can transactivate the promoter of the P-glycoprotein, also promoting the overexpression of the mechanism that provokes the drug resistance of these cells. We can monitor easily the drug resistance. You can turn up the lights, please. Thank you. And we can easily monitor the presence of drug-resistant cells, mainly by using a fluorescence microscope and using some other drugs, such as topotecan and mitoxantrone, which are fluorescent. And we have different models in our laboratory for overexpressing the ABCG2 transport. And as you can see here, wild-type cells, which lacks the ABCG2 transporter, are unable to efflux this molecule, and they will accumulate the drug into the cells, and they will die. But resistant cells are non-fluorescent, and only by using contrast microscopy and fluorescence microscopy you can describe if the vast majority of cells are able to efflux or are resistant if the vast majority of cells are resistant or are sensitive to the chemotherapy that we are using for treatment. If we can do this using fluorescent molecules and fluorescent microscopy, we can apply this technology for flow cytometric measurements, and we can incubate our cells in multiple well plates in presence of absence of different multi-drug reversal agents such as cyclosporine, PCCX333, and verapamil, which are specific for the P-glycoprotein, probenecid, which is specific for MRP, a different multi-drug transporter, or frumitremorgen C, which is specific for the ABCG22. Then, by removing the drug and removing the inhibitors, we can measure the different wells in the flow cytometer, and we can resort to multicolor substrates for drug resistance assessment. Rhodamine 1 to 3 is one of them, which emits, can be excited with the blue and emits in the green. And if we have a multi-drug resistant pattern in our cells, the fluorescence will decrease over time, as you can see here. But you can apply a different molecules, such as cyto13, which also can be excited and emits, such as rhodamine 1 to 3. But we can use some drugs, some chemotherapeutic agents, such as doxorubicin, daunorubicin. We can use also the LDS751 for monitor drug resistance and other drug that can be excited with a red cell lyser. And this is a very nice different substrates that can be very nicely combined for immunophenotyping analysis to measure specifically which subpopulation are expressing this multi-drug resistance phenotype. But cancer or leukemic cells, they don't want to die, and they can express not only in a specific multi-drug transporter. This is an example that we observed in our laboratory, showing that hepatic cancer stem cells can express 2 different multi-drug transporters. And this is a very nice experiment showing how calcein, which is well transported by MRP, but also is well transported by P-glycoprotein, will produce a different pattern when we block these cells in the presence, which prevents it, which is specific for MRP, and cyclosporin A, which is specific for the P-glycoprotein. Thus, in the absence of a molecular analysis, we are able to, in less than 1 hour or in 2 hours, that these cells are expressing MRP and the P-glycoprotein multi-drug transporter. And this information can be very relevant for the clinics. In addition, time is a very powerful parameter in flow cytometry, and we can look at the expression at the transport capacity of the different wild type or different mutants. We have these 2 mutants in our laboratory, and we can monitor the efflux of mitochondrion, which is a drug, a fluorescent drug, and we can monitor the fluorescence over time, and we always resort to study the 50% of fluorescence after over time to compare the different velocities of the different transporters. And this is also crucial to understand how the cells can efflux very quickly, or at the lower velocities the drug from the cell. And this is also crucial to understand how the drug effects will have effect or not. Not only we can measure the expression of multi-drug transporters or the activity of multi-drug transporters using functional flow cytometry, but there is a nice antibody against the P-glycoprotein that can be used also for functional analysis of the P-glycoprotein, because this molecule can suffer some conformational changes during transport. But this is a case of a acute myeloid leukemia with a huge amount of CD34 non-normal cells. These are blastic cells that can express different levels of P-glycoprotein, and we can monitor and we can add this marker to try to learn more about the malignant phenotype or the drug resistance of these cells. And then in addition, we can combine this with the fluorescence activity using some other substrates such as rhodamine 1,2,3 or Cyt13 and other markers. Well, this is just to remember that the P-glycoprotein is expressed in CD34 positive cells, but the cytopopulation cells, which can be normal but also cancer-like stem cells, will express the ABCG2 transporter. The site population is an important hallmark for the definition of the stem cell compartment, and only can detect it and isolate it by flow cytometry. These cells are CD34 negative and were discovered using ultraviolet excitation by the efflux of the H33342 dye. Now we can use a violet laser excitation to study the efflux of the dicycle violet, and we can mimic the same profile that we obtain for the site population using the classical UV excitation and compare our valid results using the violet excitation and dicycle violet. And this population can be distinguished by dual wavelength emission, because ones bind to the DNA, the H can emit in red and blue, and also for the dicycle violet, which also emits in red and blue. And this efflux can be blocked in the presence of a BC inhibitor, such as fumitremorgine C, or verapamil, which is more specific for the P-glycoprotein at higher concentrations. And they represent only a small fraction, not only of the bone marrow, because site population can be found in multiple tissues, not also in humans, but also in other species. This is the classical pattern of the site population. These are a cord blood specimen. And as you can see here, this is a linear scale, and we can see the blue emission. By combining the blue emission and the red emission, we are identifying mainly G0 and G1 cells. These are mainly S cell phase, and here we can find G2M phase because the hERG is also staining the DNA, but cells that are able to efflux because of the expression of the ABCG2 transporter will have this specific site distribution pattern, and this is the main reason because this population is called site population. And the more primitive site population cells are located in the tip of the site population because they express the larger amounts of the ABCG2 transporter. And of course, we can sort viable cells to perform different experiments. And if we are studying at the site population, this is a case and specific for a leukemia with a myelodysplastic syndrome with an increased number of malignant site population cells to show how we can block this efflux by using the specific inhibitors, and this should be used to discriminate our population. If not, we can detect easily multiple artifacts. And this is to explain these chromatic shifts in the fluorescence. This was done using thymocytes, which lacks the expression of the ABCG2 transporter, and they were loaded with the hERG dye at these concentrations from 10 to 13, and decreasing concentrations showing how this fluorescence is varying from the red to the blue. And this will mimic the classical site population pattern, showing that this can be associated to or correlated with this concentration from 10 to 10 micromolar, and this is 0.1 micromolar. And this is very important because sample manipulation can heavily affect the site population because we are performing a functional assessment of this multi-drug transporter, and this is the effect of ammonium chloride because it's inducing apoptosis, and by resorting to some density gradients, we can observe that the distribution, there is a dual tail for the site population showing that the transporter is not functioning as should be normally in normal cells. And in addition, this is to complicate the issue because human specimens can have different site population distributions, and this is enriched in very primitive site population. This is bone marrow very enriched in very primitive site population cells, and this lacks these more primitive site population cells. And this is to show how the stains is, this procedure is done. This is a saturation experiment, not this a transport or drug efflux experiment, and this is a 2-hour incubation for human specimens and 90 minutes for other species, but all the people should perform and should verify this experiment in their lab. And finally, this tail disappears in the presence of an inhibitor. And here you can see an animation of the whole staining pattern. And this was done using leukemic cells, and we believe that this site population can be enriched in leukemic stem cells. And as you can see here, cells undergoing cell division can also efflux the hERG dye, showing that they also express the ABCG2 transporter. However, in some other models, they are highly expressing the ABCG2 transporter, and as you can see here, all the cells appear as site population and cells, and we need to monitor also, or to understand more how this population are dealing with this transporter. I told you previously that chemotherapy can deplete the more sensitive cells, and we can enrich after different cycles in heavily drug-resistant cells, but also in cancer stem cells. And this is a classical, I'm sorry, this is not a classical, this is a classical profile of the cell population that appears duplicated that can appear duplicated in some leukemias. And we had the suspicion that maybe this population can be enriched in leukemic stem cells. The main reason for this dual site population, or this dual tail, is explained, but if we are staining the DNA and we are looking at the presence of the ABCG2 transporter, by using the classical propylium iodide stain, we can distinguish in this acute myeloid leukemia diploid cells and tetraploid cells. And here you can see the cell, the G2M phase for the tetraploid population, which explain that these two populations, the tetraploid and the diploid population, are effluxing the huge dye, and they are maybe enriched in leukemic stem cells. In fact, the existence of site population cells will be associated with a more drug-resistant profiles when you are looking to leukemic or cancer stem cells. In addition, we have looked at the existence of circulating site population cells, and using leukapheresis products, we were able, for the first time, in our laboratory, for the first time, to identify a tiny population, a rare site population cells. But when we simultaneously stain these cells, we observe that most of cells, near the 40% of these cells, express the CD34 positive marker, indicating that these are not normal site population cells. And these cells are leukemic stem cells that are aberrantly expressing the CD34 positive marker, or maybe some mutations that can mimic this site population profile can appear when you are looking with, or when you are dealing with very rare cells. In addition, we have developed some experiments using xenograft models, and this was done using human cells that were obtained from a prostate cancer. And we were very interested in the existence or the presence of site population cells in the tumor, but we failed to identify this population in the tumor. However, when we look at the primary cultures of bone marrow, we were able to identify a very rare population, which is uncommon in bone marrow. Most of cells should be hematopoietic cells, and as you can see here, once these cells appears as diploid, this appears as a tetraploid, because they have two nuclei. This cell also has two nuclei. And then we decide to perform some cytospins in our laboratory to study the cytokeratin expression by using this unspecific antibody against one of these cytokeratins. This was the cytokeratin egg, showing that these rare cells, these large cells appearing in the bone marrow, have a specific staining for this cytokeratin, indicating that there is a human origin of these cells that are mining the bone marrow. And when we compare a mice that was never in contact with human prostate cell cancer cells from this model, and we look at the existence of by flashing their femora in this model, we observed that most of cells were in the bone marrow, and most of these cells were from a human origin, indicating that these cells maybe are migrating by a simple tactism of the oxygen concentration, because ABCG2 also provokes resistance to the hypoxia, and this is one of the main reasons because we are very interested in hypoxia. But as you can see here, there is a high infiltration of these models. When we flush the femur and we inject again these cells in new models, these cells also migrate to the bone marrow, because the bone marrow is a very hypoxic niche, and we believe that the mice is not a good model to study this population, because these cells maybe can be dormant for a long time, and the mice is not a good model to study the kinetics of these populations. In addition, we are looking at the microRNA expression, which are co-regulated in site population cells. I will not explain more about that, but we are also interested in the site population in chronic myelogenous leukemia, which is a very, very rare population, but this site population, because of the expression of the ABCG2 transporter, is also involved in drug resistance, because the ABCG2 is able to efflux Gleevec and some other tyrosine kinase inhibitors. And this is very difficult to identify or to sort these cells to work and to obtain valid conclusions. And this is the main reason, because we would like to induce the ABCG2 transporter under hypoxic conditions. And in addition, we have studied for the first time different pathologies based on the expression on the site population, based on the numbers of the site population in these different pathologies, indicating that MDS, multiple myelodysplastic syndromes with progression, are highly correlated with the number of SP cells and the exitus of these patients. Dicycle violet can also be used to distinguish very primitive progenitors, and this was published, I think, that 2 years ago. And this is to combine the vibrant dicycle violet, which is also effluxed by the P glycoprotein, and we can monitor dicycle violet high, which are more differentiated progenitors, where compared with the dicycle violet low, because they have the highest levels of P glycoprotein. And this is in a specific case of an acute myeloid leukemia, and we can see here low CD34 positive cells with lower amounts of P glycoprotein when compared with dicycle violet low, which are more indifferentiated and brighter CD34 progenitors when compared with this plastic population. As I told previously, ABCG2 transporter can be overexpressed under hypoxic conditions, and using different solid tumor models, we were able to identify the site population, and we also look at the alkaline phosphatase under expression under this concentration. This is the atmospheric and usual concentration for most of cell tissue incubators. And under hypoxic conditions, we can see that the site population is mainly expressing alkaline phosphatase at high levels. And we have additional experiments to demonstrate that this is a very nice marker to distinguish very primitive progenitor cells. The effect of the hypoxia is very relevant, especially when you look at the whole genome RNA expression. And under this is a model of cells maintained under 21% oxygen conditions, and the same cells under 5% oxygen conditions. And when you look at the expression, 50% of the genes near 15,000 are overexpressed under normoxic or 21% oxygen conditions, and only few genes are overexpressed under hypoxic conditions. We have sorted the site population of these different models, and we have compared the gene expression profile of the whole genome, when I'm sorry, under 21% and 5% oxygen conditions, showing that again, the 50% of cells overexpress multiple genes under 21% oxygen conditions, and this will affect dramatically the phenotypic profile of the cells that we are looking for. As you can see here, if we can monitor the gene expression profile of this population, which represent in this case less than 1% of cells, we need to resort continuously these cells to obtain highly purified concentrations of these cells, and we obtain it highly purified concentrations up to the 80% of site population cells to perform the gene expression analysis and to validate these results and compare the different experiments in the two previous slides. But also, we are interested in the principle. At the beginning of my talk, I was saying that ABCG transporter and this on the pig glycoprotein and some other multi-drug transporters can also act as the firewalls or some regulators for the transduction signaling. And then we were very interested in the hedgehog pathway, and when we looked at the expression of the ABCG22 in unsorted cells, the patch expression, which has these two genes and the SmoothNet are very relevant for the regulation of the hedgehog signaling pathway. And when we look at the expression of these genes under 3 different models, we can observe an inverse relationship between the ABCG2 transporter and ESMO, and not with patch, suggesting that maybe ABCG2 is regulating the ESMO gene expression. And by using functional flow cytometry and a specific molecule, a chemotherapy drug which is called cyclopamine, we perform some competitive experiments showing that cyclopamine is also not killing the site population. These cells were only incubated with the HDI. These cells were incubated with cyclopamine, showing a dramatic reduction of the site population. And by combining cyclopamine and temozolomide, a drug also used for gliomas and astrocytomas, and the levels of the site population are near the same, suggesting that ABCG2 is affecting at this level, at the cell membrane level, and or protecting the classical sonic hedgehog pathway by avoiding the binding of some drugs such as cyclopamine, which is a target of the SmoothNet product, and then at this level, ABCG2 is effluxing cyclopamine just to avoid to stop or to interrupt the normal progression of the sonic hedgehog pathway. Using different inhibitors involved in different molecular pathways, which are very relevant for the stemness of cells, such as the Wnt pathway, the beta-catenin pathway, the Notch pathway. We have observed that most of molecules used for signal transduction experiments to block the different signaling and transduction pathways are effluxed by the ABCG transporters, suggesting that is our hypothesis that the most primitive cells express the high levels of multi-drug transporters, and this expression is aimed to regulate or to preserve the stemness of the stem cell compartment by avoiding the differentiation on the loss of the stemness of this program. If you are interested in more details about the flow cytometry of the site population, you can consult this specific chapter published in current protocols in cytometry. And this is just to finish my talk, just to show you how leukemias are highly heterogeneous in terms of phenotypes. This is just only to summarize these are different patients, And we can see here the different staining pattern for the CD34 positive and CD45 positive cells. And these are different patients showing that this is a classical expression such as can be found in cell line models. And we need to study or we need to learn more about the stem cells using other approaches, and we are using the approach of the alkaline phosphatase to study the activity between the CD34 positive compartments, showing that the unique population can be subdivided in 4 different compartments or in 3 different compartments. These are different patients. And we can look also at the expression of small CD34 positive events and large CD34 positive events. And we are working to demonstrate that these cells are very primitive. And this is a very recent results that we obtained in the last year, because we were able to monitor a case that was a acute leukemia patient that was diagnosed in May 6 years ago and died very recently, and we were able to monitor the last year based on the alkaline phosphatase activity experiments. And this is to show how, this is in one of these relapses in May 1 year ago, to show how there is a high alkaline phosphatase population which also could express the CD34 positive marker, and after treatment this population also remains, and following a new relapse this population also appeared again and was able to survive in bone marrow and peripheral blood. And this is the bone marrow analysis following a new relapse, and this is when the patient finally died, showing that this population is persistent and can help to identify very primitive progenitors, and we believe that this can be a very nice marker to study the very primitive progenitor cells. In addition, this is to show how the classical flow cytometry is unable to explain this heterogeneous pattern when we observed using the alkaline phosphatase staining, and this is the CD10 versus the CD34 and the CD19 versus the CD34, showing that all populations are apparently clonal populations, but based on the expression of the alkaline phosphatase, we can distinguish these different subsets of CD34 positive cells. This is a different case, a diagnosis and relapse, and this patient died in 3 months approximately, showing that the existence of this alkaline phosphatase overexpressing CD34 positive cells, and this is a population. Then we believe that it can be also enriched in leukemic stem samples. And this is a case when we look at from a child with Burkitt lymphoma, showing the bone marrow and the classical, using the classical experiment, using the alkaline phosphatase substrate, showing that CD10 and CD19 cells can be also very heterogeneous based on the alkaline phosphatase activity, but when we looked at the ascites, we were also able to identify these highly active alkaline phosphatase cells present and infiltrating the ascites. Just to finish, just to show that the immunophenotyping of leukemia can be very complex. With five colors will be associated with 15 combinations, and most of hematology laboratories are using 8-color combinations. But it's more better when we look at minimal residual disease, and we are dealing with small sample volumes or poor samples because of the cells are these patients can be are suffering leukopenia. We are unable to to get the enough number of cells to distinguish the positive or the minimal residual disease, but we can resort to different systems or different approaches using potent softwares that offer the possibility to transform these tiny plots or these rare events in more understandable and perform a good color compensation to obtain the better possible results when we are looking at very rare cells. And this is fundamental to understand all this work. Okay? And thank you for your attention. And I'll be happy to discuss some points.
Any question for Jordi? One.
You mentioned at the beginning of your talk.
Wait, we need to.
Very interesting data, thank you.
Thank you.
You mentioned at the beginning of your talk that one should go absolute, and in some of your plots you have really very tiny amount of cells, and you mentioned only percentages.
Yeah.
My question to you would be how many events you need to acquire in order to make, let's say it's a real population, what you see?
Yeah.
That would be my question.
This will depend on the population are you looking for at the presence of the cells or the kind of cells that you are looking for. If you are looking for cells, this is the statistics, if you are looking for populations that are less than 0.01% of cells, then you will obtain a huge number of cells. If not, sampling will affect heavily the number of cells that you are studying. The last results I'm showing about the immunophenotyping studies were done by the people at the hematology lab, and they always resort to study the number of cells using percentages, because this is the cutoff that they need to diagnose that there is a relapse or the minimal residual disease is persistent or not. But I think that we need to change this thinking to the absolute counts, because our sample manipulation can heavily affect the percentage, which is a relative number in comparison with the absolute numbers.
If I can add one thing, this is the field which I will present the last day, which is rare events, actually. So there are some rules to follow. I don't want to anticipate my talk, but there are very strict rules that you have to follow that you cannot use, actually, because if you had to apply Poisson statistics in such cases, you need to acquire maybe 100 million cells or 1 billion cells to have decent numbers to respect the Poisson statistics. Of course, this is not feasible for logical reasons. I mean, you cannot take a patient, put the patient into the flow cytometer as such. This is the only way to do this, actually. So there are other tricks that I will explain you in a couple of days.
Yeah, the experiments that we have performed using the alkaline phosphatase experiment in leukemia are usually performing using less than 100 microliters of blood, because all this sample is needed for karyotyping, immunophenotyping, for molecular analysis, and we receive only a small sample, very tiny sample.
Well, because we have a very good ethical committee, so you can use 100 microliters. Here, maybe we can use 1 microliter.
Yeah.
Enrico?
So actually, it's good that somebody raised the problem of how many cells, because I was going to discuss this. I think it's a very important point. So people tend to ask this question all the time. So how many cells is enough? How many cells to see a positive event? Or well, obviously, as Andrea pointed out, so the Poisson is important to determine this. But let's say it's not. There's no number of cells that you have to reach to define positivity. So if you acquire, let's say, a million event, a million CD8, and you find one cell that is positive for a tetramer, for example, and that cell is antigen specific, it's one cell in a million. That doesn't mean that it's not positive. It's positive, but of course, is there a biological significance in that? Maybe so, maybe not. So that is the question.
Yeah, this raises
So
another important topic about the expression of a single molecule. Maybe the future of cytometry will be able to detect a single molecule, but this single molecule can be translated into a pathological model. This is, or dim expression when compared with a single molecule of mRNA can be enough to provoke a leukemic event or not. This is a
right. So
very amazing question.
Yeah, exactly. So I don't think so. I think this conception of like analyzing at least 50 cells, at least 100 cells has been abandoned pretty much. Now I don't know if people still use it, but if you have the controls, if you excluded all the dead cells, if you can overlay, for example, a very rare event onto, let's say, the total of CD8 or the total of CD4 or whatever, and you're confident that that cell is positive, and obviously you need controls, then you can say it's positive. Then maybe it doesn't have a meaning, a biological meaning, but it doesn't mean it's not. It's not positive.
Yeah, I fully agree.
So remember, I just came from a meeting where they talked about TCRs, and apparently a single, as you pointed out, a single PMHC molecule is able to activate a single TCR, and that's enough for T cell activation. So I think everything is relative.
Mm-hmm. Thank you.
That's a great news, because in the old times we needed to activate 3,000 TCRs when I had your age. So I'm happy to know that TCR changed and there is an evolution in the TCR.
I would know how can you distinguish the debris from the site population?
The debris from the site population?
Yes.
It will depend of the conditions that you are using on your flow cytometer. And I haven't explained that, but we are dealing with different excitation and laser power that can be very helpful to distinguish debris from site population cells. Of course, cells that are effluxed, all the Hughes dye or the dye cycle violet can be pretty near from the debris, but you should monitor always by heavily following the rules or the recipe for the site population to obtain very reproducible experiments. And this is the main reason, because we are able to distinguish site population cells from debris. But if you are failing to adjust the real concentration of cells with a real concentration of dicycle violet or the Hughes dye, this will heavily affect the number of site population cells that you will identify in your experiment. And this can be heavily associated with most of cells appear as a negative site population cells, but this was related to a variation in the concentration of the drug and the number of cells.
Yeah, you've made elegant use of the site population technique, the spectral shift technique for Herxt, but it's always a good idea to remind ourselves that that dye was originally invented as an anti-helminthic drug. It's a drug in its own right. It's a DNA binding drug, and of course the spectral shift technique depends on the rate at which the molecule binds to DNA and therefore donates energy to its neighborhood molecules within the minor groove, and therefore it's not a linear relationship between for example, the function of the transporter versus what you measure on the spectral shift technique. Mm-hmm. And as a result, there are other complications, as you know, that if you use certain drugs that compete with the molecule on DNA, for example, that becomes a quenching effect, mm-hmm. which will spectrally shift what you see as well. So there are some places that the spectral shift technique cannot be used, and especially if you're using it, for example, in looking at competing drugs in those subpopulations. Even though it's a very elegant technique, as Enrique said this morning, you have to be very careful in terms of mixing probes together because they can interfere with each other. You have experience of that, with, for example, with doxorubicin treated samples.
Yeah, when I research to study the site population, I always try to avoid fluorescent drugs, but of course it is not possible always. And it is clear that there's a high interaction between multiple drugs, and also if you one problem that can arise also if you are looking for the reactive oxygen species, because if you are looking at the reactive oxygen species or you're using some other molecules, and your cancer cells or your leukemic stem cells are expressing these multi-drug transporters, and they are actively effluxing the drugs or the probes that you are using for your purpose in your experiment, you will have a high deviation in the results. In our experience, we prefer to resort to competitive experiments, and we also monitor the huge efflux in the blue when we have highly purified cells, and we use non-fluorescent drugs that are usually that can be used to block the signaling transaction for different pathways, but also using the blue emission and using non-fluorescent drugs. If not, you will have a lot of interferences. Unfortunately, we lack a good marker, a different good marker for the site population, and it is only possible to identify this population using the H or the dicycle violet. Dicycle violet is a very exciting molecule that mimics practically the site population profile and offers new possibilities because you can use less expensive lasers, such as violet lasers, and this offers new possibilities for the clinics because most of clinical flow cytometers are equipped with violet lasers, and we need to learn more about the relevance of the site population for stem cell transplantation. And
one question, you show some data on the gene expression profile, right? Yeah. Is there any molecule that you find there and you don't find in other? When you show the data on the gene expression, did you find any molecule which is only expressed by those cells?
A lot of molecules. Yeah.
And what about using?
I need to work more but this is a huge task.
No, because this could be a system to identify them as
and very unrelated molecules. Yeah. Mm-hmm. And we have proteomic results, we have microRNA results, and we have gene expression results, and there is a impressive variation in the pathways that are controlled if you are looking only at the proteomic, which is unrelated to the microRNA results and is also unrelated to the gene expression profile, indicating that maybe other regulating mechanisms can act. Yeah.
Okay. Any other question? Are we happy with the site population? Yes. Okay.
Thank you very much.
Thank you again.